Welcome to the Medical Nursing Podcast, the go-to show for vet nurses and technicians passionate about internal medicine. I'm Laura Jones, a registered vet nurse, all-around medical nursing nerd, and your guide to breaking down complex medical diseases into simple, actionable chunks. Each episode is packed with knowledge and tips that you can start using right away to give even better patient care. So let's get started. Hello, and welcome back to another episode of the podcast, or welcome if you are tuning in today for the first time. You are listening to episode 117 of the Medical Nursing Podcast, where we turn complex medical conditions into the need-to-know information so that you can spend more time caring for your patients and use more of your skills in the process. Now, at the time of recording this, I have just returned from International Cat Care's World Feline Congress, where I was not just speaking, but more importantly, learning from loads of phenomenal speakers. It was honestly fantastic. And I had a microbiome episode planned already. It was actually going to be the episode after this, but there were loads of talks on it this weekend. And after watching those, I knew I needed to sit down and talk about it now. Because the truth is, this is a fascinating and really state-of-the-art area of veterinary medicine, and it's one that I think we play a big role in as nurses and technicians. So over the next half an hour or so, that is exactly what we're going to look at. We'll explore what we know about the microbiome so far, so what it is, what affects it, and also how it impacts way more than just the GI tract, and also what our role is in microbiome medicine, so that you can care for your GI patients physically and emotionally and use more of your skills in the process. So with all of that said, let's get started. So the first thing that we're going to look at is what the microbiome is and why it is so important. And before we start to look at things in detail, I do quickly want to explain a couple of terms because you will hear the microbiome called different things depending on where you look. So there are actually two terms. We've got the microbiota and the microbiome. The microbiota is the community of living organisms, so it's the actual bugs. And while this is the case, to be technically correct, the microbiome is slightly different. The microbiome is the collective genome. And by this, I mean that it's all of the genes that all of those organisms, the bugs themselves, carry. To be honest, most of the time we use the term microbiome as a bit of a catch-all term, and some people will actually use the two terms interchangeably. So today I'm going to be using the term microbiome. That's what I generally say as a kind of umbrella term, but I do just want to point out that there is a technical difference between the two. So with all of that said, put simply, the microbiome is a collection of microorganisms, so bacteria, yeasts, fungi, protozoa, viruses, etc. And we actually have several distinct microbiomes. So we have the one in our GI tract, which is what we are predominantly talking about today. But alongside this, we also have a microbiome on our skin, in our respiratory system, and also in places like the urogenital system as well. And when we look at the role of the microbiome, we do need to just touch on the different ways that microorganisms exist either in or on a host. So there are three terms that we need to be aware of. Microorganisms can either be what we call commensal, symbiotic, or pathogenic. So commensal microorganisms are where that microorganism or microorganisms benefits without harming or helping the host. So basically, the commensal organism kind of eats at the same table as the host without causing any trouble. A symbiotic relationship is where there is a mutually beneficial relationship between both the microorganism and the host. So for example, if we look at the GI tract as an example of this, some species of bacteria within the GI tract will receive nourishment from things like sugars in our food. And as they break those sugars down to receive that nutrition, that helps us to digest those nutrients better because the bacteria have started to break them down for us. And that's a symbiotic relationship. Then we have a pathogenic relationship, and this is pretty much what it says on the tin. So this is where our microorganism benefits, but in the process of them benefiting from the host, they harm the host in some way, causing things like infection, damage, or disease. So this is what we are treating in our unwell patients. In the microbiome, the microorganisms should be either commensal or symbiotic, so not harming or directly benefiting the host, and we should have low to no numbers or well-controlled numbers of pathogenic species. We'll look at dysbiosis in more detail later on in the episode, but as an overall kind of summary, dysbiosis is a disruption in the balance of these microorganisms. So this could mean that there are reduced numbers of certain microorganisms or a change in the balance between those beneficial and pathogenic species. In dogs and cats, bacteria make up around 98% of the GI microbiome, and there are a lot of them. In fact, there are around 100 trillion microbial cells within the intestine, and that is up to 10 times more actual cells we have in the entire body. So technically, and this was a great fact I learned from one of the sessions at iCat Care this weekend, we and our patients are more bacteria than we are human, dog or cat or other species, depending on the patient we are talking about. And then when you count genes, the microbial genes outnumber the host's own genes by something like 10 to 1. So actually, we could argue that we should think of any animal as kind of like a multi-species hybrid, because we've got the host cells and then the microbial cells in their microbiomes working together as one big ecosystem. So each microbiome is a distinct community which is shaped by its own little environment. Current research suggests that we all, and that I say we as humans and animals, begin as sterile beings. So we have no microbiome at all. And then during parturition, when that human or puppy or kitten is born, they will actually start to pick up their first microorganisms from the vaginal, oral and skin communities of the mother. So those maternal microbiomes are literally going to start feeding that gut and beginning that individual's GI microbiome. Looking at the gut microbiome in more detail, the number and the diversity of those microorganisms increase as we move from the stomach down to the colon. The stomach actually holds relatively few bacteria. It has some things like Helicobacter and also some bacteria that produce lactic acid. But because it's an acidic, hostile place, and it's supposed to be, right, because the stomach is supposed to break down food, it doesn't really house many bacteria because not many of them will thrive in that environment. The small intestine has a mix of aerobic and facultative anaerobic bacteria, so species like Lactobacillus and Streptococcus. And then as we get further down into the colon, we get a really, really full, busy and diverse microbiome. So this is where we have lots of anaerobic bacterial species like Clostridium, Fusobacteria and Bacteroides. So I don't want to get too bogged down in the different types of bacteria because there are so many of them, but I do just want to say the main bacterial groups across dogs and cats are pretty consistent, and they include things like Proteobacteria, Bacteroides, Firmicutes, Fusobacteria and Actinobacteria, and I have probably butchered all of those names. While we will see varying proportions of each of these in between individuals, we know that we do see these big kind of blanket categories of bacterial species pretty consistently across our patients. And then the next thing that we need to look at is what actually affects the composition of the GI microbiome, because there are a lot of things that can impact it. And the biggest factors in cats and dogs are thought to be diet, environment, the age and developmental stage of the patient, alongside things like genetics, medications, and health status. And I want to touch on a few of these in more detail. So firstly, looking at diet, in multiple studies, diet has been identified as the primary influence over both the structure and the function of the GI microbiome over short and long periods of time. So we know that diets that are higher in protein and fat tend to increase levels of certain bacteria like E. coli, Clostridium perfringens, and also Fusobacteria. Diets that are higher in fibre will actually feed the fibre fermenting bacteria within the gut. So this is species like Faecalibacterium, Megamonas, and also Prevotella. And these bacterial species are fantastic because they actually produce metabolites which are known as postbiotics. And these bacterial metabolites actually help the gut, they keep the gut healthy and they enrich it. Also, the digestibility of the diet matters, because if we have poorly digestible diets, they actually can disrupt the microbiome further. So studies have shown that poorly digestible protein and fat sources can disrupt the microbiome. I do also want to mention here that dogs and cats are not the same. We know that our dogs and our cats have different nutrient needs, and also their microbiomes will respond differently. In fact, some studies have shown that the feline microbiome will respond more quickly to things like dietary changes than dogs. But I don't want to get too bogged down in how each individual species responds. The main thing we need to consider is that cats and dogs will respond differently. And we can't assume, therefore, that they are all going to have the same response to how we modify the microbiome, feed a certain diet, administer antibiotics, etc., etc. Next up, we need to look at genetics. So the microbiome, much like our patients, is continually evolving. As we said, it begins at birth through the maternal skin and vaginal flora, and it is then shaped after birth by things like maternal grooming, lactation, and then that individual puppy or kitten's environment. Early on in that patient's life, it will be highly variable. If we think about things like maternally derived antibodies, for example, they come and go, and it's going to take time for that microbiome to stabilise into a mature colony or community of beneficial microorganisms. We also need to consider that lots of the milestones that our neonatal patients move through as they grow, like weaning, for example, will all have significant effects on the microbiome. So in the very early stages, for example, before a patient is weaned, environment and mum's microbiome is the biggest factor that impacts that individual's microbiome. Until after weaning, once the patient begins eating quote unquote proper food, diet will then take over as the biggest factor impacting their individual microbiome. Lastly, we need to talk about medications, because these can also significantly impact the GI microbiome. And out of medications, antibiotics are probably the most obvious category for us to talk about, because we know that they have significant and long-lasting effects on the GI tract. They can actually reduce bacterial diversity and balance within the gastrointestinal tract, and this change can persist for a long time. In fact, in one study, the use of tylosin, which is a common antibiotic used in GI disease, caused a persistent drop in bacterial diversity in about 40% of dogs. Also, other medications such as antacids like omeprazole can also alter the microbiome, although we know that these changes tend to be shorter lasting and typically they will resolve themselves when that medication is stopped. And this is also important for us to consider, because this explains why we have really moved away from routine use of antacids in recent years, and also why we can often see things like diarrhoea with omeprazole use. So it shouldn't routinely be a drug that we reach for in gastrointestinal patients unless there is things like ulceration or GI hemorrhage. I also want to talk about how we assess the GI microbiome, because sometimes we will do this, and also when it's needed. Now, I don't want to make this a really deep scientific episode because I feel like it's quite brain heavy. This is all very state-of-the-art stuff that we're talking about. And I want to make sure that it's practical for all of us as veterinary nurses and technicians. But with that said, it is important that we mention how we assess the microbiome and also how we have come to learn so much about it in recent years. So until recently, culture was the only way that we could assess bacteria. So we would take a sample, we would incubate it on an agar plate, and we would see what grows. The problem with this is that most of our GI bacteria are anaerobic, so they don't use oxygen for cellular respiration. They thrive in the absence of oxygen. And this means that in standard lab conditions, which is an oxygen-rich environment, they won't grow. So we couldn't explore 90+% of the microbiome until very, very recently. Now, don't get me wrong, I am not throwing out culture as a diagnostic that we use in GI disease. It definitely has its place. We use it routinely to detect things like Salmonella and Campylobacter, for example, in patients with diarrhoea. It's just not suitable to look at the entire GI flora. Now, thanks to things like bacterial DNA sequencing, we have a much better idea of just how much is going on inside the gut. We also have tests like FISH, which is fluorescence in situ hybridization, and this test allows us to identify different bacteria inside GI tissue. And we will use this quite commonly where we suspect there is invasive bacteria causing GI disease. We will send biopsies away for FISH, and the different bacteria will actually fluoresce when you have a look at them in the lab. I also want to mention something that you will probably have heard of with the evolving chat about the microbiome, and that is a test called the dysbiosis index. This is probably the test that is performed most commonly in practice to look at the microbiome, and it is a panel of PCR tests which looks at key bacteria and turns all of those different bacterial species into a single result that our vets can interpret. So we will often use this to quantify changes to the microbiome, either in a patient with GI disease where it's needed, or when we are screening faecal donors. And all of these different testing methods have actually taught us a few things about the microbiome. The first being that every individual is different. Each patient carries a unique community of microorganisms with its own proportions of the major groups. Despite this, though, the genes that these microorganisms carry are similar, which is why patients can have changes in the particular makeup of their microbiome, but those different organisms carry out the same functions. These highly individualised changes also explain why patients don't respond to many GI interventions in the same way, so different diets, different probiotics, etc. won't reliably have the same effect across individual patients. The second thing that we need to mention is that measuring the microbiome is challenging, and it's also not without limitations, which is why we don't do this super commonly in most GI patients. If we think about what impacts the microbiome, it's a lot. In humans, the microbiome is known to be affected by so many things like diet, sleep, environment, when a person eats, so the gaps between meals, medication, exercise, hydration, and much more. So if that microbiome is ever changing, measuring it only really gives us a small snapshot of a much bigger picture. Additionally, there's also no standardisation between different laboratories, so the same sample can give different answers depending on how the DNA was extracted and analysed. And that's why we don't routinely assess this in all GI patients. Also, we know we need to look at more than just the microbiome, and I'm air quoting right now because the microbiome we know is huge. But what I mean by this is the GI tract will often tell us there is a problem through systemic signs or wider blood work changes. We know this because we run all of those blood work changes in our GI patients. So dysbiosis, for example, often affects things like B12 and folate levels. Because the pathogenic bacteria within the gut will basically kind of eat the B12 and use it as an energy source, and they will excrete folate. So we know, for example, that a low B12 in the face of a high folate level is highly suggestive of dysbiosis. So with all of that said, we need to ask ourselves, is there always a need to perform dysbiosis index testing if we're already highly suspicious of dysbiosis? And these are all things that our clinicians need to consider when they are working up a patient with GI disease. This episode is brought to you by the Foundation Award in Medical Nursing. The Foundation Award is my very own 12-month on-demand course designed specifically for veterinary nurses and technicians who want to feel genuinely confident nursing their medical patients. If you've ever felt overwhelmed by a complicated case, or like you're just ticking boxes on a hospital sheet rather than truly understanding what's going on with your patient, then this course is for you. The Foundation Award goes beyond just throwing information at you. It gives you a real framework for turning your knowledge into hands-on, patient-centred nursing care that really uses your skills. You'll work through it in your own time with live case-based support built in, so you are never learning on your own. If you're ready to go from feeling stuck to leading care with confidence, then head on over to the website and check out the Foundation Award. You can find the link in the show notes. And now, back to the episode. I mentioned that the microbiome is so much bigger than just the GI tract, and what I really want to talk about now is something that's really important, and that is the link between the microbiome and disease. This is an essential area for us to discuss because, as we've said, the microbiome plays a bigger role in all disease in general in our patients than I think we have previously appreciated. If we look at what the microbiome does, it is responsible for, or it contributes to, tons of different physiological processes within the body. This includes supporting metabolism and energy use. It includes creating vitamins like B vitamins and vitamin K. The bacteria within the GI tract also will compete for space and nutrients with pathogenic bacteria, and naturally this then controls the level of pathogenic bacteria within the body, and it helps to keep that balance. It also regulates the immune system and much, much more. So we can see that the role of the microbiome exists way past just the GI tract, and this means also that dysbiosis has a bigger impact on our patients than just within the gut. In fact, alterations to the microbiome have been linked with chronic enteropathy, CKD, obesity, diabetes, skin diseases like atopic dermatitis, and even immune-mediated disease. We don't have a good lot of data on this in veterinary patients, but in humans, also neurodevelopmental and mental health conditions such as Alzheimer's and depression are all highly associated with changes to the microbiome. So if we look at veterinary patients and the link between the microbiome and certain diseases, studies have shown that obese patients have an altered microbiome with lower populations of energy harvesting microorganisms than lean cats. Again, in cats, there is a link between intestinal lymphoma and dysbiosis. So studies suggest that chronic dysbiosis causes inflammation, and this inflammation in turn changes the environment and can increase the risk of tumour formation. And another area that we are learning more and more about is the link between the GI tract and the brain, and also how dysbiosis therefore impacts the emotional health of our patients. So if we look at what we call the gut-brain axis, there are both neurochemical and anatomical links in between the gut and the brain. So the two body systems communicate via the bloodstream and also the nervous system through the vagus nerve, which is one of our cranial nerves that supplies the GI tract, and also through neurotransmitters as well. And this means that the gut and the brain are more heavily linked than we have previously thought. And this is an essential consideration for us as veterinary nurses and technicians. Studies that have looked at germ-free mice, which are mice that were raised with no microbiome whatsoever, showed that they were generally more anxious, less social, and had exaggerated stress responses. However, if their microbiome was recolonised early on, these changes actually resolved. But if it was left too late, some of these changes became permanent. So it seems like in early years, the microbiome helps the developing brain. In fact, around 90% of the body's serotonin is made and stored in the gut because we have bacteria in there that convert tryptophan in the diet into serotonin. Other bacteria within the microbiome produce GABA, which is an important neurotransmitter, and others are involved in the production of things like dopamine. So we know that the role between the gut and our emotional health is really important. And along these lines, we also know that stress impacts the gut because it's bi-directional communication. The gut and the brain are constantly talking to each other. So stress will cause the release of things like cortisol and other stress hormones, and these can actually weaken the GI barrier and negatively impact the microbiome, predisposing our patients to dysbiosis. So this means a stressed patient can develop GI disease, but also because we have got that bi-directional communication between the gut and the brain, GI disease can also manifest as emotional or behavioural signs rather than obvious gastrointestinal signs. In fact, one study showed that cats with inflammatory GI disease showed increased levels of anxiety and attention-seeking or care-soliciting behaviour than healthy cats. And it was actually the same for cats with inflammatory skin disease. For us as veterinary nurses, this is really, really important because we need to be looking beyond those obvious or traditional signs of GI disease that we have all become accustomed to looking for. We want to be asking our clients about things like the home environment, looking for subtle signs of stress or other changes that could be impacting that gut-brain axis. We need to be on the lookout for signs like pica, toileting or appetite changes, repetitive licking or over-grooming, and it's also the same in dogs. Many dogs who compulsively lick surfaces, for example, or even lick the air, have been found to have underlying GI disease, and often treating that underlying GI disease will resolve those behaviours. So that's the importance of the microbiome and its role in disease, but let's chat a bit more now about what happens when it goes wrong. So we have touched on what dysbiosis is, but in reality it is way, way bigger than just a change in the microbiome. Dysbiosis means that there has been a change in either the makeup or the diversity of that GI microbiota that affects how it functions. And as well as being a change in those microorganisms, it's also a change in the beneficial effects that come with them. It's a change in the metabolites that they produce, it's a change in their genetic makeup, and all of this basically means it's a weakening in the intestinal barrier, either in one area of the GI tract specifically or throughout multiple areas. There are usually 3 types of dysbiosis, so it can be defined as either a loss of the beneficial bacteria, a drop in the overall diversity of bacterial species within the gut, or overgrowth of those pathogenic bacteria, and these 3 types will often overlap in the same patient. Other factors that have also been suggested in impacting dysbiosis include the presence of abnormal substrates within the gut, so if there's lots of undigested food, for example, because this can drive metabolites which cause diarrhea. Also, if there's an increase in total bacterial numbers, especially in the small intestine, these can actually produce more toxic metabolites that have an adverse effect on the gut. And also, if we have an increase in the invasive species of bacteria, these can actually adhere to the intestinal mucosa and cause inflammatory changes in the GI wall. And of course, we also need to look at how underlying GI disease is linked to dysbiosis as well. In chronic inflammatory enteropathy, for example, we end up almost in a bit of a vicious cycle where the intestinal mucosa is remodeled because of chronic inflammation, and this chronic inflammation then impacts absorption. When absorption is impaired, we have undigested food sitting in the intestinal lumen, and this feeds bacterial overgrowth, which drives more dysbiosis, and so on and so forth. The cycle continues. So speaking of dysbiosis, what signs do we see and how do we manage it? Well, the clinical signs really are variable because it depends on the location and the severity of the dysbiosis, and also whether there is an underlying gastrointestinal disease. Patients may be asymptomatic, or we may see varying signs like vomiting, diarrhea, inappetence, and weight loss, and this could be acute or chronic. Usually patients with acute diarrhea have more mild microbiome changes compared to patients who have chronic enteropathy. Acute patients also tend to have self-limiting changes to their microbiome, and that's really interesting because if we think about current evidence, it is recommended that we do not use antibiotics in acute diarrhea because actually we will worsen the dysbiosis rather than significantly improve the diarrhea because we know it's often a self-limiting change. Treating dysbiosis is actually multimodal, including things like diet, probiotics, and also other treatments that we're going to touch on like FMT. Starting with diet, highly digestible diets reduce the amount of undigested nutrients within the gut and therefore the availability of food for bacteria, meaning it's harder for them to overgrow. Just like any other GI patient, if a food sensitivity is suspected, then we should be thinking about using an appropriate hydrolyzed or novel protein diet, just like we discussed last week in episode 116, where we looked at chronic enteropathy. Fermentable fibre sources are also fantastic in patients with dysbiosis because they are converted by bacteria in the gut into short-chain fatty acids. These fatty acids feed the epithelium in the gut and they also lower the pH within the intestinal lumen. And this pH change makes it harder for pathogens to thrive. So they have a direct kind of defensive effect on the GI tract. Speaking of those fermentable fibres, we also need to talk about pre- and probiotics. So prebiotics are non-digestible fibres like fructooligosaccharides or FOS and also psyllium. These act as food for the beneficial bacteria in the gut and they boost production of those short-chain fatty acids. Probiotics, on the other hand, are the actual live beneficial organisms and they are really, really strain specific. We know now there are like 100 trillion different species and different bacterial strains, and each of those produce different kind of DNA. So it's such a diverse population within the GI tract, we need to know the strains of the probiotics we are using. So make sure if you are using probiotics for the management of a specific condition, for example, that there is evidence showing that the particular strain of bacteria in your probiotic has been studied and is beneficial. Then we've also got faecal microbiota transplantation, or FMT. An FMT is a growing procedure in small animal GI disease, and it's also one which we are ideally placed to run as veterinary nurses and technicians. The concept is exactly what it sounds like. You take faeces from a healthy donor and you transfer it into the GI tract of a diseased recipient with the aim of restoring a healthy microbiome. Looking at how FMT is performed, fresh faeces is collected from an appropriate donor, blended with saline and strained to make a liquid solution. This solution can then be administered either rectally as a retention enema or endoscopically instilled directly into the GI tract. You can also get lyophilized oral capsules depending on your location. I personally have no experience with these because I'm based in the UK where they're generally not easy to source, but I have heard from colleagues that they're quite large and sometimes this can make at-home administration very challenging for clients. When we talk about FMT, one of the most important things to talk about is selecting an appropriate donor. This is actually probably more important than we have previously considered, given what we now know about the role of the microbiome in things like behaviour and also obesity. And selecting a donor and advising on this is also one of the biggest areas that we are involved in as nurses and technicians. So we need our donors to be clinically healthy, they need to have a normal body condition score, and they need to be emotionally healthy, so no increased anxiety, aggression, etc., etc. They shouldn't have any history of chronic GI signs, and they should have had a dysbiosis index performed and this be documented normal. They need to undergo extensive infectious disease testing and test negative for this, and they need to have received no antibiotics for at least 6 months, and they should not be on a raw diet or have received raw food within the last month. They should also be rescreened regularly, at least every 6 months. FMT is a safe, well-tolerated and minimally invasive procedure, and it can be performed by us as nurses and technicians under direction of the vet, so it's a fantastic use of our skills. Speaking of our role as nurses and technicians, I want to talk now about how we can use more of our skills to support dysbiosis patients. And if we think about where our role fits in working up GI disease, it's huge. History taking, for example, is really, really important here, and it's often a nurse-led thing. We need to be looking at things like diet, recent medications, GI signs, and now, after everything we've chatted about today, also behaviour as well. Every single visit, we should be asking about toileting patterns and faecal scoring using a standardised chart like the Purina system, and again, we are ideally placed to do this as nurses. Where further GI interventions are needed, like endoscopy, for example, which we will discuss in a separate episode, our role is huge. I won't repeat too much here because we did discuss this last week in episode 116, but things like anaesthesia support, endoscopy assistance and sample handling, for example, are really important. Speaking of sampling, there's also sample collection and handling for dysbiosis index testing, client education around diet, pre and probiotics, and monitoring response over time, including reinforcing to our clients that the microbiome takes months to recover, even when the patient looks well and even when their diarrhoea has stopped. We mentioned last week that expectation management and support are essential in GI disease to help with compliance, especially with things like diet trials, and that's also true for dysbiosis patients as well. Antimicrobial stewardship is another big area for us as nurses and technicians. We have seen today that antibiotics often aren't the answer in acute diarrhoea and that they come with significant consequences to the microbiome. And yes, while we are not going to be prescribing as vet nurses, we are often the ones having these conversations with clients, explaining why diet and supportive care are preferred and why antibiotics are usually not the answer. And then there's FMT itself. We can and should be the ones preparing the product, running and coordinating a donor screening program, administering the FMT, and looking after both our donors and also our recipients. And as FMT becomes more widely performed, this is a real area that we can champion as veterinary nurses. So let's sum up everything we have discussed in today's episode because it has been a big one, actually one of the biggest and heaviest deep dives I think we've done in a while, and there has been a lot of science we have chatted about today. So if you take anything away from this episode, let it be this 2-minute summary. The microbiome is a vast, individual and dynamic ecosystem that does a lot more than just help with digesting food. It trains the immune system, it makes metabolites which help maintain the GI barrier, and also it's in that constant bidirectional communication with the brain, impacting emotional health as well as gut health. Dysbiosis is a disruption in the balance within this ecosystem, and it occurs for many reasons, so dietary changes, environmental changes, underlying disease, and even those emotional changes that we've just mentioned. We manage it through things like diet, pre and probiotics, and faecal transplantation where needed, and avoid the use of antibiotics routinely, instead using them only where it's specifically indicated because we know that they have long-lasting effects on the microbiome. And of course, we are ideally placed to help with all of this as veterinary nurses and technicians, from initial diagnostics to FMT, to emotional health and stress management, and much, much more. So don't underestimate the importance of our role in the care of these patients. That is everything I have for you today in this deep dive on the microbiome. I hope this has been helpful and has given you some new ways to think about how we approach GI disease in practice, especially around the emotional health, which I think is really, really interesting. If you've enjoyed the episode or found it useful, please don't forget to share it with a colleague, pop it in your practice WhatsApp chat, send it to someone who will also find it useful, and let me know what you have thought of the episode and how often you see things like stress behaviour or emotional changes in patients with GI disease, because I would be really, really interested to know. Pop me a message on Instagram, let's have a chat about it, because I am really keen to know more about the patients that you are seeing and the signs that you are seeing in them. Thanks so much, as always, for being here and for choosing to learn with me. I can't wait to speak to you again about GI disease in a future episode very, very soon. Bye. If you've made it this far, then thank you so much for listening all the way to the end of the episode. And I have one more thing to ask of you before you go today. I would love to hear from you. Yes, you. I love hearing about the cases that you're seeing, the skills that you're using, and the conditions that you want to learn more about. Drop me a DM on Instagram. I'm at Vet Internal Medicine Nursing, and don't be shy. Come on over, say hello, and tell me a bit more about you. Talk to you soon.
Your love is one of a kind. Your ring should be also. Veragio diamond engagement rings are designed with European elegance, unlike any other ring. Mervis Diamond Importers is the premier source for Veragio bridal rings and for the world's most brilliant diamonds, because your forever deserves extraordinary beauty. Our experts will guide you to the perfect engagement ring paired with a diamond that sparkles forever. Your ring should be as unique as your love story. Mervis Diamond Importers for Veragio rings and world-class diamonds. See us on mervisdiamond.com.